we introduced photosynthesis when we had our photosynthesis lab, and we said it was a two-step process. It's right there in the name, photo and then synthesis. The first part is our light-dependent reaction. We're going to need to know where it takes place, what are the things that go into this reaction, and what are the things that come out. And then the second step is the synthesis part, where we actually build a larger molecule. We're going to see where that occurs and what are the things that go into and come out of this second step, the synthesis part of photosynthesis. So maybe before I list these locations, let me attempt to get a basic sketch on the board. And what I'm going to draw is a chloroplast. It's the organelle of photosynthesis. And I went and grabbed this model because it shows us an example of a cell that has chloroplast. We typically think of plants when we think of chloroplast, but they don't have a monopoly on these chloroplasts. We're going to see them in prokaryotes. Cyanobacteria have chloroplast. We're going to see the single-celled euglena, the organisms in the pond water that were swimming around on their own. They have chloroplast. And these chloroplasts work the same, whether they're in a euglena or in a plant cell. So I'm going to take up as much space as I can here. Really large chloroplast. And that means everything outside of this chloroplast, like we can see in this cell, is just the fluid of the cell. Let's write that in this color. We described the fluid of the cell as cytoplasm. So there's the cytoplasm, and that means the fluid inside the chloroplast is going to have to get a different name other than the cytoplasm. We're going to call it the stroma. And then floating around inside the stroma are these discs. We call them thylakoid discs, and I'm going to draw an exaggerated thylakoid disc. Hopefully that's enough space. Let's label this thing our thylakoid disc. There's a fluid pocket inside this thylakoid disc. Let's just call it the thylakoid space. We doing good so far on that? All right. Let's add to that then. I'm going to put a couple of protein structures embedded in this thylakoid disc. This is the membrane of the thylakoid disc, and we're going to see, I'll put them as these big rectangular structures. These are going to be photosystems, and I'm just going to label them PS1. This is what we call photosystem 1. And this is going to be photosystem 2. They both are able to capture wavelengths of sunlight, but they do different things. So I'm going to put a little asterisk next to the inside of this one. So we'll switch to this color. We'll put a little asterisk to the inside of photosystem 2, and then I'm going to put a little asterisk to the outside of photosystem 1. And we're going to learn that photosystem 2 is going to be the first one involved in this light-dependent reaction. It turns out that this one was discovered first, so it's called photosystem 1. But after photosynthesis was better understood, it turns out that upstream of photosystem 1, there's another photosystem. So being discovered second, it's called photosystem 2. But we're going to see it's going to be the first one involved in this process of the light-dependent reaction. The only other protein I want to add is going to be down here. We've seen it before. It looks like a little upside-down tree. And we call this our ATP synthase. All right, so we've set up some membranes. We've labeled the spaces in between there. I'll talk about what's going on between each of those things. But let's practice maybe first some membrane transport stuff. We know in the cytoplasm, the fluid that this chloroplast is floating around in, there's a lot of water molecules out here. So I'm just going to put some water molecules surrounding this chloroplast. And it's taken me a second, but now I've got so many water molecules out here that they're in really high concentration in the cytoplasm, and there's not many water molecules inside this chloroplast itself. We know that water molecules like to move from an area of high concentration towards an area of low concentration. We said water molecules can cross that membrane all by themselves, or if they want to, if there's an additional aquaporin, they could travel through one of these proteins. But anytime we see water moving across a membrane from high to low concentration, what do we call this? What type of membrane transport are we looking at? Yes, let's call this osmosis. We see it's passive because water molecules are always going down their concentration gradient from high to low concentration. So we're going to see they're going to end up diffusing into this thylakoid space. So we can imagine some water molecules reaching equilibrium inside the stroma. Put a few out here. Okay, so osmosis has taken place. We've got water crossing this membrane and has reached equilibrium. That's going to be important because we're about to zoom into our first step here, this light-dependent reaction. We need to see where it takes place and what are some of the things that go into it. And we've put enough on the board right now where we can answer the first part of that question, which is the light-dependent reaction is going to take place on the thylakoid disc. Let's list the things that go into it. We're going to see 4 things that we're going to list. I'm looking for the right color. We're going to use, I'll use the orange color to represent the first reactant, which is going to be sunlight. Sunlight is going to be needed to split a water molecule. So we're going to put sunlight, we're going to put water in there. The other 2 reactants, the things that we're going to add to this in just a second, let's put this dead battery of what we called ADP and this extra little phosphate that has been ripped off of it. We described this structure as like a rechargeable battery that's just dead. It's exhausted of all of its energy. So you would have to plug it back up to the wall and recharge it if you're going to get any work out of it. So there's ADP, this rechargeable battery that's dead. And then we've got this other molecule that we're going to call NADP with a little plus sign. We're going to see this as an electron and hydrogen carrier. So more on those in a second. Let's maybe see where those things come in. Let's walk over to this thylakoid disc. And I'm going to add a pretty sun into the corner of this diagram, and we're going to see UV rays coming from the sun. And what's interesting about these photosystems is that they are capable, we're going to look inside of them in a second, and we're going to see they contain pigments that are capable of capturing certain wavelengths of sunlight. What's different about photosystem 2 versus photosystem 1, here is a wavelength of sunlight that's about to do something that only happens on photosystem 2. And we're about to get a chance to do another type of membrane transport. Water molecules that venture too close to this side of photosystem 2 are going to get hit with a photon of sunlight. And what happens is these water molecules are going to get ripped apart. We practiced this last time. We know that if we have 2 water molecules and then we use this photon of sunlight to rip the water molecule apart, we know what's coming out of that. Each water molecule has 2 hydrogens. So there's 1 water molecule busted up. Here's the other 2 hydrogens from that second water molecule. Each water molecule is carrying an oxygen. So there's 1 oxygen from the first water molecule. There's the second oxygen from the other. Those 2 oxygen atoms are going to combine and form oxygen gas. We've got a couple of things happening here. These hydrogen ions, they have a charge to them. And as long as the sunlight is out, you can expect that more water molecules are going to have this same fate. They're going to be busted apart. So in the daytime, this thylakoid disk is filling up with hydrogen ions because we're splitting water molecules. The other thing that's happening is that oxygen gas molecules are being formed as we split those water molecules. So we kind of get a buildup of oxygen gas, and we get a buildup of these hydrogen ions inside that thylakoid disk. Here's my question. How is this oxygen gas going to get out of this thylakoid disk? What membrane transport is going to carry it across the membrane? What did we say about oxygen gas? It was oxygen and carbon dioxide. They were two gases that behaved the same way in how they crossed a membrane. It is passive, so there's going to be no energy required in getting these molecules to go from high to low concentration. It is simple diffusion, that's right. Why is it simple diffusion? Well, it's diffusion, obviously, because they're going from high to low concentration. But what made it simple diffusion?
It doesn't need something to help it move.
There's no protein channel. It can just go right across that membrane all by itself. Okay, so this we said simple diffusion. But what about those hydrogen ions? Those are tiny, but they have a charge to them. Remember, the membrane separates based off of size and charge. These are tiny, and they don't have a charge, so they can just cross by simple diffusion. Those hydrogen ions are tiny, but they have a charge. So what they're going to have to do, the only way out is through this ATP synthase. We'll just put it there. So hydrogen ions are going to be flowing from this thylakoid space out through the ATP synthase. The stroma, we're going to see, always has a low concentration of hydrogen ions. Compared to the thylakoid space, this is always going to have a very high concentration of those hydrogen ions. The reason the concentration, at least one reason the concentration is so high, is because when the sunlight is out, we can see water molecules being busted up, and this provides the constant influx of hydrogen ions. Okay, so we see hydrogen moving across this ATP synthase. What type of membrane transport is that? Facilitated diffusion, that's right. Let's somehow label that in there. So facilitated diffusion happening right there where those hydrogen ions are using that protein facilitator to cross the membrane from high to low concentration. You know, I should have used this analogy. I was thinking the other day, there's a shop vac that I bought not too long ago. You know, you plug it up to the wall. It's like a big vacuum with a hose. And they make some of those shop vacs that can suck up water in case you have a big mess and this floor is covered in water. Imagine if I had that shop vac, right, and I sucked up all the water from this carpet, I would maybe have to have that thing plugged up to the wall for 15 minutes or so, but eventually I would have expended enough energy to get all of those water molecules inside this shop vac bucket. But then I could unplug it from the wall, and with no energy at all, right, if I just kicked that bucket over, the water would again go from high to low concentration with no energy added. I can do that without it being plugged up to the wall. But the second I want to go against the concentration gradient, when I want to get all of these water molecules that are already spread out and put them back into a condensed bucket, well, now I got to plug it back up to the wall. We need energy to go against the concentration gradient. So back to this. So far, we're not going against the concentration gradient at all. So we're going down the concentration gradient, either water crossing the membrane on its own, tiny nonpolar molecules crossing the membrane with no help of a protein, or tiny charged particles crossing the membrane with the help of a protein. This is a good example of membrane transport, and what we're looking at, I forgot to label at the very beginning, is this is the first step of our light-dependent reaction. I'll maybe just label it here since we've already got it on the board. On the thylakoid disc is where this light-dependent reaction takes place. So it happens on the thylakoid disc with these photosystems as long as the sunlight is out and there's water to split. So this kind of sets up this imbalance between a lot of hydrogen on the inside of the membrane and very little hydrogen on the outside. What I think is so fascinating about this is the same way that we have figured out how to create these hydroelectric power plants by getting water to flow across a little wheel that spins. Water's going from high to low concentration. It's these hydrogens that are flowing from high to low concentration and generating ATP for this cell. This ATP synthase, just to finish that off, here we go. We said every time hydrogens are flowing across this ATP synthase, it's taking the two little pieces, ADP and the extra little phosphate, and sticking them together to make a larger ATP molecule. All right. The last thing that we need to say about this light-dependent reaction is something about this NADP+ molecule. We can see where water is getting used. Sunlight is splitting the water molecule right on that photosystem. And then we can see from this large concentration of hydrogen ions, they can only flow out through that ATP synthase. So that spins that like a little wheel and generates ATP. So you can see how we go from using this ADP molecule, and what's going to come out, one of the products of that is going to be ATP. So we've listed one product. The other product that we're going to see, well, just to list, I skipped over what happened with the water molecule. We saw that in a water molecule, we basically broke this thing apart into hydrogens, electrons, and oxygen molecules. Oxygen gas just diffuses right out. As soon as it's made, it's tiny, it's nonpolar, it can diffuse out of the chloroplast. So what's left behind from water is the oxygen, and then we know that those hydrogens and those electrons are about to participate in, at least for the hydrogens, they're going to be moving across this ATP synthase. I'm reminded, you're good? The electron stuff too? Yeah. I'm reminded that I haven't mentioned at all these electrons, so I guess that was timely. I think I asked last time, and I've already kind of given it away because I've said the word electron, but I was asking what is the glue that holds those hydrogens and oxygens together. When they were locked up in a water molecule, they were sharing electrons, right? And we've just ripped apart these water molecules, so those electrons are also going to come out. What's the name that we gave to these hydrogens and these electrons that come off of breaking apart these chemical bonds? They are free radicals. And we're going to see that too many of them would be bad, but we need them in order to do certain processes, like manufacture ATP. And let's see where these electrons go. As soon as the water molecule is split up, we're going to see electrons are going to be pulled into these photosystems. We're going to catch up with all this stuff on the slides, but I think it's helpful to draw this first. We're going to see the photosystems they have pigments in them that are capable of capturing these wavelengths of sunlight. And what the sunlight does when it hits these pigments, it excites these electrons from a lower ground state to a higher energy state. Maybe think of it this way. When they're originally coming off of this water molecule, it's like they just woke up. They're tired. They don't want to do anything. But the photons of light in this photosystem are going to give them a pep talk. It's like a pep rally for these electrons. They get all fired up by the time they're out of it, and they're at this really high energy state at the top of this photosystem. Between these two photosystems, we're going to see a staircase. I'll draw it like this. Not a perfect staircase, but it works. This staircase we're going to call an electron transport chain. Sometimes just abbreviated ETC. Sorry, I can't break that. ETC. Electron transport chains are always going to work the same way. This is the first time we've encountered an electron transport chain, but we're going to see them when we get into mitochondria as well. These electron transport chains, they are like a staircase, and what happens is electrons that are fed into the top of this electron transport chain, they fall down the staircase all the way down to the bottom, just like a slinky would down a staircase. I don't know if that's even a commercial anymore. Has anybody seen those commercials of the little slinkies? It's just a spring. It's not going to walk to the top of the staircase by itself. You have to physically pick it up and carry it to the top of the staircase. But if you can carry it up to the top of the staircase like these electrons, then you can just set it in motion and that slinky will fall down the staircase on its own. Then it's down to the bottom of the staircase, and the only way that you're going to get it to go down the staircase again is to excite it up. Here's our next photosystem. It too can capture wavelengths of light, and electrons that land in this photosystem 1 initially are exhausted. They're in a low energy state. But as they spend time in this photosystem, sunlight's hitting them, they're getting pumped up. They're eventually going to get brought all the way back up to this high energy state. And going off of photosystem 2, there's another electron transport chain over here, but it's a shorter one. I'll just label this electron transport chain. This second electron transport chain, when electrons are excited up to the top of the staircase and then sent down this electron transport chain, they don't end up at another photosystem. Instead, their fate is to end up being picked up by this molecule, NADP with a little plus. This swoops in. It picks up those electrons. It actually picks up a couple of hydrogens and is converted into this molecule, NADPH. This happens outside in the stroma, away from that thylakoid disc. But you can see the final product. This NADP with the little plus sign is going to come out as NADPH. It's picked up electrons. It's picked up hydrogens. So it no longer has that charge, and we write it with an H. But now we've got everything, the location, the reactants, and the products that come out of this light-dependent reaction. Oxygen, we've checked that off. The hydrogens and those electrons, we can see the electrons being funneled into this electron transport chain, eventually picked up by this NADP+. The hydrogens, they're stuck going through this ATP synthase, and then they flow out into the stroma. One last thing to say about this light-dependent reaction, and then we'll move out to the stroma. That is, I haven't mentioned what kind of work is being done by these electrons in motion. Anytime electrons are in motion, I think I said last time, if you stick a paperclip into this light socket, you'll feel electrons in motion. They can be used to do work, like power the lights or charge your cell phone. Here is electrons in motion, and they're going to be used to do work. And here's maybe a better representation of the electron transport chain. When we catch up with our slides in a second, we're going to see these electron transport chains aren't really like a staircase, but instead they're a series of proteins that are embedded in the membrane between these photosystems. And what they do is, as long as electrons are in motion, what's going to happen is that they are grabbing hydrogen ions that happen to flow out here in the stroma. Let's follow one of these hydrogen ions out through the ATP synthase. It's out here in the stroma. There's a relatively low concentration of hydrogen ions out here compared to how many there are inside. So it's easy to see that these hydrogen ions are going to flow out. But as long as electrons are in motion down this electron transport chain, what they do is they grab hydrogens from an area of low concentration and they throw them back into this thylakoid space. That's going against the concentration gradient. That's going to require energy. That's like me trying to shot back up the water that's on the floor. If we're going from a spread out low concentration towards a high concentration, we're going to need to plug it up to the wall. So hydrogens are only going to move against their concentration gradient when there is energy doing this. So those moving electrons provide the energy. This is our only example of active transport, ions going against their concentration gradient. If the sun goes down, then this whole chain of electrons shuts off, and there is no more active transport of hydrogen ions in. This is why this is the light-dependent reaction. It'll all be running when the leaves are getting hit by sunlight, but it shuts down at night. Let's see. Okay, we've ripped apart our water molecule. We've got our oxygen gas that's diffused out. We can see what's happening with those electrons and hydrogens. We've made our ATP by sticking a phosphate onto an ADP, that little wheel that turns the ATP synthase. And then we've loaded up these electrons and hydrogens, and we're about to go out into the stroma. Just so that I start off labeling this, out here in the stroma is where we see the second step of this process, what we're going to call the light-independent reaction. This light-independent reaction is also in some textbooks referred to as the Calvin cycle. I'll use both of them. You can recognize both of those as being the light-independent reaction. We need to know the same thing, the location, and then what are the things that go into and what are the things that come out of this light-independent reaction. And I've given the first one away. Where this occurs is in the stroma. And at least part of these reactants are already there for us. They are the products of the light-dependent reaction. We're going to see these two things are complementary. The light-independent reaction is going to use some of the products from the light-dependent reaction. The products that it uses are the ATP. Let me get my ATP color. So the things that go into this light-independent reaction, we've got our ATP molecules ready. We're going to end up using 6 of those ATP molecules. The other thing we're going to use are the hydrogens and the electrons that are currently loaded up in this NADPH molecule. So we start with NADPH. And the other thing that we're going to start with are carbon dioxide molecules. There's going to be 6 carbon dioxide molecules. And I feel like we've reviewed this already. If not, it's a good chance to review anabolic reactions. Remember, in anabolic reactions, we start with small things, these tiny carbon dioxide molecules. And through this process of the light-independent reaction, we're going to stick these 6 carbon dioxides together to build a big glucose molecule. Anytime we're going from small to large, we did this last time, it's going to require, it's like building a puzzle, right? There's going to be some things that need to go in. We said some of the things that need to go into forming these new chemical bonds are the glue, the electrons, and the hydrogens. That's being provided by the NADPH. It gives up its electrons, it gives up some of its hydrogens, and those are going to go into sticking these carbons together to form more bonds. So those are the glue, and when it gives up its electrons and hydrogens, you can see it goes back to its original form, this NADP+. It's sent back to the thylakoid disc, and it's going to pick up more electrons and hydrogens right here. So that's recycled. The other thing that's going to be recycled down here is the ATP. We also said in addition to the glue, the electrons and hydrogens, we're going to need energy to build a puzzle. I still have that 1,000-piece puzzle that I have not put together because I don't have enough energy to do so. But we can see this ATP molecule being broken down into ADP and that individual phosphate. Again, ATP is being used up in order to stick these small carbon dioxides together to build a larger glucose. I said this is a good example of an anabolic reaction because we're going from small to large. Anytime we're going from small to large, it requires electrons, it requires energy. So endergonic. What else? Let's go over here and see. We've got our location. We've got the reactants, the things that are going into this. So you can see by the time we're done with the light-independent reaction, this ATP molecule has been broken back down into the ADP and that phosphate. This comes out of the Calvin cycle, but it's the same thing that's about to be used up in the light-dependent reaction. We've just used up the electrons and hydrogens from the NADPH, so it goes back down to NADP with a little plus. But that's okay. That's the products of the Calvin cycle are the reactants of the light-dependent reaction. And the final thing that we have built there is our glucose molecule. We've gone from small to large, and we're only going to be doing that if we're using up energy and using up those electrons and hydrogens. Okay, so whether you say it by listing these reactants and listing the products, or you sketch something like this and then describe where these two reactions are occurring, either one will work. But there's only one more thing I want to add to this Calvin cycle. It's a substance that is waiting right in here called rubisco. This rubisco is a substance that the carbon dioxide molecules are going to combine with. Maybe I'll just draw it this way. Carbon dioxide is going to combine temporarily with this molecule called rubisco on its way to being built into a glucose molecule. We'll catch up with the slides because I won't be able to draw that accurately in this little amount of space. And the other thing that's going to be a bummer when I pull down the screen is it's going to block half of the diagram, but I'll leave it up. We don't have to draw anything else today. In fact, the slides that we're about to click through are the same slides of this process. And unfortunately, the screen is blocking half of what we have drawn already, but we do have our reminder chart of the things that go into and the things that come out of each of these reactions. So let's see some more detailed pictures in the slides of what we just kind of broke down, and it's a nice little review. I brought that model in here, but what this picture shows is that when we think about photosynthesis, I think at least me, the first thing that comes to my mind are plants. And these plants have a green color due to the presence of that chloroplast. But like I was trying to mention with that cell, plants don't have a monopoly on photosynthesis. They're not the only ones with chloroplast. There's the euglena. We saw a few of those moving around under the microscope in our pond water. They too have chloroplast. This is a seaweed. It's a brown algae. It has chloroplast. It's capable of photosynthesis. And here are chains of cyanobacteria. They're prokaryotic cells, but they too are capable of photosynthesis. So plants have it, but again, we see it in other organisms as well. If you have chloroplast, then you are autotrophic. You can make your own glucose molecules as long as there is sunlight and water available. So we put in all the proteins in the membranes and talked about the membrane transport that led to the production of that ATP and eventually building the glucose. Let's look more closely at this chloroplast. When we did our photosynthesis lab and we were looking at how that worked in leaves, we may have looked at this picture where in order for photosynthesis to occur in a leaf, there are these little openings on the underside of a leaf that we called stomata. I think I compared this to the word stroma. It sounds very similar, but they're different things. Stomata, these are openings on the underside of a leaf that allow gas exchange to occur. So whenever we inhale oxygen and then breathe out carbon dioxide, plants are doing the opposite. They're inhaling carbon dioxide and exhaling oxygen. And it happens right through those little openings called stoma. Inside the leaf itself, there is a middle layer of cells called the mesophyll. This is the layer of cells where photosynthesis occurs because those are the cells that contain chloroplast. Here we are zooming in on this chloroplast, and better than I've drawn on the board, it's back. All right, let's say that's not helpful. Better than I've drawn on the board, we can see this chloroplast is a double membraned organelle. So that outer membrane is made of the same thing, this phospholipid bilayer. And then we can see the inner membrane is going to have these folds that form these discs. We're going to call those thylakoid discs. You can see an entire stack of those thylakoid discs is called a granum. I didn't draw a granum, I just drew one of these thylakoid discs and we zoomed in on it so that we can see the proteins embedded in it. So we're going to end up zooming into one of those thylakoid discs to see this light-dependent reaction. I put this up on the board so we know the overall chemical equation for photosynthesis. Not that, I would say remember the top one more than the actual number of carbon dioxides or hydrogens that go into this. We know that we're going to take tiny carbon dioxide and water molecules, and we're going to end up with these larger sugar molecules. Oxygen is a byproduct we saw from ripping apart water molecules. But we're going from small to large, and the only way we're going to go from small to large is if there is an input of energy. We said that comes from the sunlight. Over here, if you look at the actual number of water molecules, it is true that the light-independent reaction, the second step, is going to end up producing 6 water molecules, but we ended up using 12 in the first step. So there's an overall loss of water molecules. You're using up 6 water molecules to build these sugars and oxygen. Okay, so we got the things that go into and come out of photosynthesis, but it's a 2-step process. We said yeah. Carbon dioxide and water are going to go to sugar and oxygen, but what you'll need to know is that this is a 2-step process, right? The first part is the part where we use the sunlight to split the water. That's where we get the oxygen. And then the second part, the synthesis part, is where we use the carbon dioxide and the ATP and the electrons to build the sugar. So carbon dioxide and sugar come from the second part. The first part, we're just splitting apart water molecules and getting oxygen and getting those hydrogens and those electrons. Setting up that flow of hydrogens like that hydroelectric power plant, getting that little ATP synthase to spin. So all the energy, all the ATP is produced by splitting those water molecules with light. Again, the hydrogens and electrons are used in that process, and oxygen is just diffusing away by simple diffusion. In the second step is where we take the carbon dioxides and stick those together and make the sugar. So first step, we said the light-dependent reaction occurs in sunlight as long as there's water available in that thylakoid disc. So water and sunlight go into this reaction, and we can see what are coming out of it. There's oxygen as a byproduct, and we can also see ATP being made and this NADPH being produced, loaded up. The second step is the Calvin cycle, this light-independent reaction. We can see it takes place out in the stroma. It uses the ATP in this NADPH that we just produced in the light-dependent reaction. These are the things that go in, and they get used up. So what comes out of this Calvin cycle, we can see the ADP, now the dead battery, in this NADP plus. It's given up its electrons and hydrogens in the Calvin cycle, stitching together these individual carbon dioxides to build a glucose. So there we're using up our carbon dioxide, and it ends up as one of these glucose molecules. Here's a larger picture of this. I think I tried to put this type of diagram on the board when we did our overview of photosynthesis, right? Because you can see what goes into and what comes out of each of these reactions. I think that I've used words to represent the same thing that we're seeing with this diagram. The light-dependent reaction, you've got water and light that go into it. You've got oxygen, NADPH, and ATP that come out. And then we can see the same thing for the Calvin cycle. The Calvin cycle is using up this ATP. It's using up our NADPH, and it's using up this carbon dioxide. And what comes out of it, we can see the NADP plus. We've got the ADP and the individual phosphate, and now we've got this glucose molecule that we've stuck together. So this is maybe a nice visual overview of these things. But what I don't like about this is that it doesn't show exactly how the light is splitting the water molecule. It doesn't show the interactions on the membrane of that thylakoid disc. So here's where I want to zoom into the thylakoid disc and look in detail at what happens when these water molecules are ripped apart. We had this sketch on the board. We know when you rip apart a water molecule, you've got oxygens. These are tiny nonpolar molecules that can just diffuse away by simple diffusion. But those hydrogens and those electrons, the other pieces of the water molecule, are going to be locked inside that thylakoid disc. The hydrogens have to flow out through this ATP synthase, and the electrons are about to get funneled up to the top of the staircase. They're going to go through the electron transport chains, eventually ending up in this bucket of NADP+. Okay. Something about light. It's crazy to think that of the entire electromagnetic spectrum, we're only able to observe with our eyeballs a thin sliver of this electromagnetic spectrum. It's known as the visible wavelengths of light. And we're going to see that plants are absorbing certain wavelengths of light, not the entire spectrum. You know, if you look at a flower, it's got all these different colors. But if you look at the photosynthetic part of a flower, that's the leaf, the green part. The leaves are going to be full of a bunch of pigments called chlorophyll A and chlorophyll B as their primary pigments. primary pigments, like I said, or trying to say, chlorophyll A and chlorophyll B. We're going to see these are little proteins that are capable, they're sometimes described as light antenna because they are tuned to certain frequencies of light. They can only pick up certain frequencies. So you can see in this diagram, chlorophyll A and chlorophyll B are absorbing. They're picking up a lot of the blue and the violet ends of the visible spectrum. So those wavelengths of light are the ones that are being absorbed and used to split apart that water molecule. In addition to the blue ends of the spectrum, we can also see a lot of the red and orange ends of the spectrum are being absorbed. There's something funny about how our light perceives colors. You see the colors that are not absorbed, right? Like this bag, I'm going to pick your red bag up real quick. We all see this, heavy one, we all see this as a red bag, and that's for some reason it's because it's absorbing all of the other wavelengths of light except for red. So when we look at it, that's the color that we see. I'll set that down gently. When we look at the leaf of a plant, it looks green to us because those are the only wavelengths of light that are not being absorbed. The ones being absorbed, like we said, the blue and the red end of the spectrum are going directly into splitting those water molecules. Here is one of those little chlorophyll A or chlorophyll B molecules, and in this sketch you can see that it is absorbing a photon of light, and what it's really doing, what this sketch is trying to show it doing, is that it is giving a little pep rally. It's giving a pep talk to these electrons that are in their tired, exhausted ground state. But this chlorophyll molecule can pass on some energy from the photon, and now this electron is fired up. It's ready to go. Put me in. I'm ready to go. So it's in its excited state, and what it's going to do is it's going to be sent from one photosystem to the next. Here's our first close-up look at one of these photosystems. I've just drawn it as a purple box, but you can see these photosystems are going to be loaded with those pigment molecules, chlorophyll A, chlorophyll B. They're tuned to picking up certain photons of light, and when hit with a photon of light, we can see they pass that energy on to electrons, kind of like a trampoline giving these electrons a boost up to the top level. Now these electrons are up to the top level. I'm reminded for a second of a picture that we saw back in our metabolism lecture. Does anybody remember that picture when there was that guy jumping off of the diving board into the water? Right, so at the top of the diving board, he's in his excited state. He only got to the top of that board because his kinetic energy took him up to the top of that diving board. These electrons initially are in their low energy state, but the energy passed to them by these photons get them up to the top of the diving board. So now they're in their high energy state, and they could, like that slinky, once it's been carried to the top of the staircase, now it can fall down. And electrons in motion can be used to do work, just like they're doing from that plug. I've talked about these two photosystems, photosystem 1 and 2. I've drawn them on the board. But here is another sketch of these two photosystems. And we automatically can see this is photosystem 2 because this is the only one that's splitting a water molecule. There's no water molecules being split, no electrons being released over here. All of the new electrons are coming from splitting the water molecules in photosystem 2. We can see once we split the water molecule, the electrons are carried into this escalator of electrons. The oxygen gas is diffusing out by simple diffusion, and we know those hydrogens are going to be stuck inside that thylakoid space. Let's follow the electrons for a second. We can see now they've received their pep talk. They're taken to the top of the staircase. In their high energy state, they just simply flow down this staircase like a slinky between photosystem 2 and end up down here in photosystem 1. Again, photosystem 1, we said, isn't going to create any new electrons, but it has those pigment molecules. So it is capable of taking these exhausted electrons and giving them a pep talk, getting them right back up to the top of the staircase. So these photosystems are going to excite electrons, and once they are excited in their energetic state, they can move down another one of these staircases. But look at how the electron transport chain coming off of photosystem 1 is a little bit shorter. It's not going to do the same thing as this electron transport chain between the two photosystems. We're going to see that the electron transport chain between the two photosystems is going to help make ATP. I kind of don't like this picture because it gives you this false idea that electrons moving down this electron transport chain immediately produce ATP. They don't immediately produce ATP. We talked about what they do. They let hydrogen ions be pulled into the thylakoid space. That is, they're being pulled against their concentration gradient, so that requires energy. That requires electrons in motion. And once we saturate this thylakoid space with hydrogen ions, the only way they can flow out is through that ATP synthase. So indirectly, these electron transport chains make ATP by keeping the concentration of hydrogen ions out here in the stroma very low. We said if hydrogen ions end up out here and wander too close to the electron transport chains, they're going to be pulled back into that thylakoid space. Okay, so it's only the flow of hydrogen ions across that ATP synthase that makes this ATP. That is helped by these electron transport chains because it pulls hydrogens into the thylakoid space. All right, I'm going to get off that thylakoid space and we're going to go to this next electron transport chain where we can see it's shorter. The electrons at the end of this staircase are picked up by NADP+. So we can see NADP+ picking up hydrogens, picking up electrons, and now it's drawn as this NADPH. Here's another way of looking at these two photosystems. Again, we start with photosystem 2. This is where the electrons come from in the first place, and this has shown a worker acting as a photon of light, exciting these electrons up to the top of the staircase. So they wouldn't get there on their own, but again, the photon of light carries them up to this high energy state, and now in this high energy state, they can flow down this electron transport chain. Remember, the first electron transport chain makes the ATP, not directly, but by keeping the flow of hydrogen ions going out into the stroma. And then the second photosystem that receives these exhausted electrons can excite them again by more photons. The pigments in these photosystems capture those photons of light and get these electrons excited. So now at the top of the second staircase, these electrons are going to go down an electron transport chain that ends up in this bucket of NADP+. Okay, so we've drawn it. We've seen it a couple of different ways on the slides. Here is now our first close-up actual picture of a thylakoid disc. You can see the photosystems, that's photosystem 2 and photosystem 1. You can see the series of proteins that are the electron transport chain. It's not exactly a staircase, but we said it's a series of these proteins. And as long as electrons are moving down those series of proteins, what's happening is hydrogens are being pulled into this thylakoid space. Remember, there's already a high concentration of those hydrogen ions inside this thylakoid space because that's where we're splitting water molecules. We can see photosystem 2 splitting water molecules, providing the flow of electrons into this electron transport chain. It also provides the water molecule. Let me try that again. It also provides the oxygen that's coming off of this water molecule, and we can see oxygen diffusing on its own. But these hydrogens are going to be stuck inside this compartment. They can only flow out through the ATP synthase. That's the only place that's generating ATP. So we can see at the end of the electron transport chain, those electrons end up at photosystem 1. They're excited once again to go down the second shorter electron transport chain that ends in this NADP+. All right, so now we've loaded up this NADP+. It's got its electrons, it's got its hydrogens. We've produced our ATP because the hydrogens have now spun this wheel enough that we've loaded up on ATP. We can see oxygen being diffused away from this thylakoid disc, so we've produced everything that we need to do for the light-dependent reaction, and now we make our way out into the stroma for the second part. I just wanted to show this slide for a second because it is a preview of something that we're going to see on the second, well, it's the third unit. We're in unit 2 now. When we come back to start unit 3, we start with the mitochondria. And we're going to see the mitochondria is also made up of membranes. There's this inner membrane, and when we look at the inner membrane of a mitochondria, we're going to see these same two things, electron transport chains and ATP synthases. And they're going to be doing the same thing in the mitochondria. We're going to see electron transport chains. When electrons are falling down them, they're going to pull hydrogen ions across the membrane towards higher concentration. The same thing happens in a mitochondria. We're going to see electron transport chains. When they are working, they pull hydrogen ions into an area of higher concentration. The dark gray areas represent high concentrations of hydrogen ions. In a chloroplast, we can see the high concentration of hydrogen ions is in this thylakoid disc. That is true, and that means those hydrogen ions have to constantly try and flow out into the stroma. And it's that flow of hydrogen ions that makes our ATP. The same type of flow of hydrogen ions across an ATP synthase is established in the mitochondria, but we're going to see the high concentration of hydrogen ions, to give it away, are in this outer perimeter space. And then hydrogen ions are going to have to flow towards the middle, this inner matrix of a mitochondria. So the direction of hydrogen flow is going to change, but it's still the same thing. As any hydrogens move across that ATP synthase, generates our ATP. In the plants, we can see it flows from the thylakoid disc into the stroma. And out there in the stroma is where we use that ATP. We use those electrons and hydrogens to stick these carbons together to make our glucose. So here in the second step is where we go from carbon dioxide to sugar. We're synthesizing something here, so we need ATP, we need the NADPH. We're going to see that most plants are called C3 plants. That just means that every time they go through one of these Calvin cycles, they're going to take in 3 of these carbon dioxides, and they build these little 3-carbon sugars. We know glucose is a 6-carbon sugar, and we're going to see the Calvin cycle produces half of a glucose molecule every cycle. 2 halves of a glucose molecule can get stuck together to build a whole glucose molecule, but building chains of carbons are versatile. You can build a bunch of different things with chains of carbons. I mentioned this rubisco is going to be a molecule that is involved in the Calvin cycle. Let's see how it is involved. Better than I have sketched on the board here, we can see rubisco, it just sits in the stroma and it waits for carbon dioxide. And what happens is you can see there are 3 carbon dioxides taken in. We'll just go 1 carbon dioxide at a time. So let's imagine 1 carbon dioxide coming in here. The oxygen is ripped off and we just have 1 carbon that gets stuck onto this 5-carbon structure. Rubisco is a 5-carbon structure. So when we add a carbon to it, we've got this 6-carbon structure. We can all add. That's 5 to 6, no big deal. This 6-carbon structure is unstable. So it quickly breaks apart into 2 of these 3-carbon structures. I know there's a lot of numbers going on here. We've got 3 of these 6-carbon structures, and when they all break in half, we get 6 of the 3-carbon structures. Okay, so let's follow it. The 3-carbon structures are going to be called these G3Ps, and we're going to see each Calvin cycle is going to produce that 3-carbon structure. Here's that G3P, the 3-carbon structure. Remember, we had 6 of those 3-carbon structures. One of them is going to end up as G3P. Then when we go from 6 and we give up 1, we're going to be left with just 5 of these 3-carbon structures. Here we are. We've now got just 5 of the 3-carbon structures left because we've given 1 of them up as the G3P. 2 of those can be stuck together to make a glucose, like we said. Or you can just use 1 of those 3-carbon structures and make a glycerol backbone. You could rip a carbon off of there and have 2 carbons to create an amino acid. So we're sticking together chains of carbons to synthesize and build stuff. These 3-carbon structures, we can see what's left over. We've got 5 of these 3-carbon structures. And what we're going to need to do is rearrange these 3-carbon structures into a 5-carbon structure. 2 things to point out here. We've got 5 of these 3-carbon structures, so 15 carbons total. And then as we rearrange these, we're going to end up with just 3 5-carbon structures. So we still got 15 carbons. All we've done is rearrange them. Specifically, we've rearranged them to go from a 3-carbon structure to a 5-carbon structure. We've gone from small to large. Anytime you're going from small to large, it requires energy to go in. And sure enough, some ATP is being used right here to synthesize this rubisco. The whole reason that we wanted to point this out is that this molecule rubisco is recycled. Each Calvin cycle, 3 carbon dioxides are used to make a 3-carbon structure. And once again, rubisco is waiting on another series of carbons to come in each Calvin cycle. So we start with rubisco, we end with rubisco in this Calvin cycle. That is the last slide Just wanted to go one to prove it to you, then I'll come back to this one. Because this was my attempt, at least on the PowerPoint slide, to write down all of the reactants and the products of both the light-dependent reaction and the light-independent reaction. We can see sunlight splits a water molecule, and it's the splitting of that water molecule that produces the oxygen gas that diffuses out, releases those hydrogen ions and electrons that are going to go into the electron transport chains or flow across that ATP synthase. That flow of hydrogen ions across that ATP synthase makes our ATP. It's helped, remember, the electron transport chain helps out that ATP synthase by removing any hydrogen ions that end up out here in the stroma. They're pulled back into that thylakoid space. And then at the end of those electron transport chains, we can see this NADP plus is converted into NADPH. Picks up electrons, it picks up hydrogens. And now we go to this Calvin cycle where we take those individual carbon dioxides, and we're going to end up building a glucose molecule out of this, but anytime we're going from small to large, we said it's going to require ATP. So we're busting up 6 ATPs down to individual ADP molecules, and we're using the electrons and hydrogens from NADPH, breaking it back down into this NADP plus. Let's see. The intermediate steps here I would not worry about. I would know that if we're thinking about the Calvin cycles, it takes place in the stroma. It is piecing together carbon dioxide and building these 3 carbon sugars. The carbon dioxide combines with this Rubisco molecule. It's regenerated through this Calvin cycle. So anytime we're going from small to large, we can see not only are we going from 3 carbon dioxides to a 3 carbon structure, ATP and electrons and hydrogens are being used up. So we need those free radicals to synthesize stuff. When we were breaking things down, like when we were breaking apart the water molecule, then it was releasing those free radicals, the electrons and hydrogens.